Inside us, cells constantly remove and recycle part of themselves. This is autophagy, a process I studied during my PhD. In this video, I'll explain how this process works, why cells depend on it, and why it matters for biology and human health. The word autophagy comes from Greek and literally means self-eating. That may sound destructive, but autophagy is actually one of the main ways a cell protects and maintains itself. Instead of allowing unwanted material to accumulate, the cell captures part of its own contents, breaks that material down, and reuses many of the resulting components. This process became widely known outside cell biology in 2016, when Yoshinori Ohsumi received the Nobel Prize in Physiology or Medicine for discovering key mechanisms of autophagy. Much of his foundational work was performed in yeast. Yeast cells are simpler than human cells, but many of their basic cellular systems are highly conserved. By studying yeast cells in which autophagy failed, Ohsumi and other researchers identified genes that control the pathway. Those discoveries gave scientists a framework for understanding autophagy in animals and humans. Every cell has a maintenance problem. Proteins can become damaged or misfolded. Organelles can stop working efficiently. Mitochondria, for example, can lose function and begin producing excessive stress signals. Even normal metabolism creates wear and tear. Over time, stress and damage generate material that the cell can no longer use safely. If this material remains inside the cell, it can interfere with normal cellular functions. So the question is, what happens to this material? Cells have several quality control systems, and autophagy is one of the most important. It acts as an internal cleanup and recycling pathway, especially when the damaged material is too large or complex for other degradation systems. Autophagy begins with capture. A membrane structure called the phagophore begins to grow around cellular material. As this membrane expands and closes, it forms a double membrane vesicle called the autophagosome. The autophagosome then fuses with a lysosome. Lysosomes contain enzymes that work in an acidic environment and can break down proteins, lipids, and damage cellular structures. After fusion, the resulting compartment is called an autolysosome. Inside it, the captured material is degraded into smaller molecules, including amino acids, fatty acids, and other cellular building blocks. These components can then return to the cell and be reused for energy, repair, or the construction of new cellular material. Sometimes autophagy captures material relatively broadly, but it can also be highly selective. For example, the selective removal of damaged mitochondria is called mitophagy. Other forms of selective autophagy can target protein aggregates invading microbes or damage parts of the endoplasmic reticulum. Here, a damaged mitochondrion is recognized and surrounded by a growing membrane. As the membrane expands, it gradually isolates the mitochondrion from the rest of the cell. Once the membrane closes, the damaged organelle is sealed inside an autophagosome. The autophagosome then fuses with a lysosome. The damaged mitochondrion is broken down, and useful molecular components are released back into the cell. So autophagy is not simply cellular destruction. It is controlled removal followed by recycling. This quality control system becomes especially important in long-lived cells such as neurons. When mitophagy is impaired, damaged mitochondria can accumulate and contribute to cellular stress and neuronal dysfunction. Defects in pathways involving proteins such as PINK1 and Parkin are linked to inherited forms of Parkinson's disease. This does not mean that reduced autophagy alone automatically causes Parkinson's disease. Neurodegeneration is much more complex, but these connections show how the failure of cellular quality control can contribute to disease. Autophagy is now one of the most active areas of modern cell biology because it connects cellular maintenance with metabolism, immunity, aging, cancer, and neurodegeneration. But the goal is not simply to increase autophagy as much as possible. It is a tightly regulated and context-dependent system. The real challenge is understanding what should be removed, in which cell and at what moment. A healthy cell is not a cell in which nothing ever goes wrong. It is a cell that can recognize damage, respond to it, and restore order. And that is why autophagy matters.